Hey guys, welcome back to Bioinformatics with R. We are in process of running our RNA-seq analysis with EdgeR. This last step for this video, it's going to look a lot like the microarray stuff because the Pathview package is applicable to any differently expressed genes that you identify. So we're going to use, instead of the Arabidopsis database that we had used for the microarray dataset, we're going to use the mouse database, and we're going to pull out some annotations and then run our Pathview. So let's get into it. So like I said earlier, we're doing this in our Markdown just because I want to show you how to integrate your images into our Markdown. So we're going to do columns of org.capital M, lowercase m, this one, eg.db. So this is the mouse database that we're going to interface with. Select lines. Okay, let's do ttglm2. Which we had from the last, which was our top table from EdgeR. Jeez. Okay, and we're gonna say it's as a data frame. ttglm table. So we're gonna pull out the table from this, and we're going to, which has our log full change and everything, and we're going to save it into a new variable called ttglm2. This doesn't seem to be running. Let me restart this session. I'm going to restart our real quick here. It off here. There we go. I probably have to rerun my libraries real quick. Okay, so we pull out the columns for this database, and then we saved this ttglm2. So we have our gene name, log full change, log counts per million, p-value, and our adjusted p-value. Okay, so now we have to do this heavy coding again, which I know sucks. Symbol equals map IDs. Remember, we're going to pull out our variables of interest, so our keys for it to use to search is going to be the row names, ttglm2, and then our column is going to equal symbol. Our key type is going to equal ensemble. So we used intres before, but we're using ensemble because we're doing a mammal here. Multivariables, we're going to use the first one. Now let's do ttglm2. Let's make another one called entres. Map IDs. We're going to do org.m.eg.db, and then we're going to say keys. So some of this I'm just going to copy and paste here. So we're going to take this stuff, copy, and we're going to paste it there because we're doing essentially the same thing, except for we're changing symbol here to Entrez ID. And then I'm just going to kind of cheat my way around this here, right? Copy that, paste it, and the last one we're going to call name, and we're going to change column. It's going to be gene name, and then we're going to call, oh, I have to get rid of these parentheses. Okay, so take a screenshot, this is what it has to look like. Well, let me run it first, I guess. See if it works. All right, so let's run this code. TTGLM 2 not found. So I ran that one. What did I change here? Or not change correctly? Shouldn't be capitalized there. Shouldn't be capitalized there. Okay, let's run this again. All right, there we go. So this is what it should look like. So now we have our gene name, log full change, count per million, p-value, adjusted p-value, the symbol we've added. So this is like the actual symbol you'd read in a paper, like I say, the Entrez ID. We got duplicate of p-value and stuff. Oh no, this is just scooching over one. So this is columns 1 through 8, and this is columns 5 through 9. So we also added this, we called it name, but it's like the actual description, inhibitor of DNA binding 3, what the actual genes do. So we've added a little context to what our differentially expressed genes are. So now let's do some Pathview. So let's load Pathview. Let's load GAGE. I'm not going to go too much into what these did because you know that from the Arabidopsis microarray stuff. So it's the same thing, right? So we're just taking our differentially expressed genes from this technique and running it through the same Pathview thing. So full changes are TTGLM to log full change. So the names of our full changes that we just created. So let me run these first few lines. Okay. So full changes is just the expression that log2 full change of everything. So for names though, we need to name these because if we look at, say we just look at head of full changes, it's just numbers, right? And it's not associated with any gene. So we have to add the names, and they're TTGLM2, and we're going to use the Entrez IDs. And the reason that is because this is what's going to, Pathview needs Entrez IDs to map. So now if we look at the head, of full changes, you see that the Entrez IDs, just these numbers here, are present for each expression though. Okay, so CAG.mm equals CAG.gsets mouse, this time instead of Arabidopsis, and then ID type equals Entrez. And then let's do CAG.mm.sigmet equals CAG.mm $kg sets CAG.mm sigmet.idx. Okay, let's run these. Okay, so if we look at CAG.mm, we have this table, nested table, right? So we have our KEGMM, and then within there we have KEGMM sets, and then we have the G name, what it kind of does, etc. So with KEGMM Sigmet, we took out the KEG sets, and from there we took out the Sigmet IDX. So let's look at this again. Sigmet IDX, which is these numbers right here, which we'll use shortly. Okay, so let's get the results. So KEG res equals GAGE, full changes, gsets equals KEGMM.sigmet, same directory equals true. Okay, so we should have KEG results, KEG res. So we have a list of 3. So we have our greater, our less, and then our statistics again. L apply, KEG res head. So this gives us, so it goes over all the pathways in here and shows us the head. So we see that we have ribosome as greater than, pluripotency of stem cells, which is interesting. Less than, we have the phagocytosome, cell cycle, necroptosis, etc. So similar to Arabidopsis, these are just mammalian pathways. Okay, so now let's do like we did before. Graders is KEG res greater. Lessers is KEG res less. So let's run those 2 lines. And so we saved those into variables. Now, let's start mapping these out. Okay, so KEG res pathways, data frame, ID is equal to row names, KEG res greater, and then KEG res greater, and let's pipe it down. TDL, make it into a table data frame. I'm going to filter row number less than or equal to, let's not do 12, let's do, we'll do 5 again. So we'll print out just the 5 tables that are from ID, as character, KEG res pathways. Okay. All right, so let's run this code. Forgot to pipe it down. Okay, so we have our top 5 pathways from greater, ribosome, pluripotency stem cells, TGF beta signaling pathway, Notch signaling pathway, Hippo signaling pathway. Now let's do KEG res IDs, and we're gonna remember we have to get rid of these annotations because Pathview doesn't like it, so we just want the numbers here. So we're going to do the substring KEG res pathways, and start equals 1, stop equals 8, and then let's print keg resids. Perfect. Got rid of them. And now let's do that plotting. Plot pathway equals, and make that function again. PID Pathview gene data equals full changes. Pathway ID equals PID. Then species equals mouse this time. My gosh, hit the right buttons. There we go. Oh, we gotta add new sig signature equals false. Okay, and then now we can use the function to plot multiple pathways. So temp is sapply keg res IDs. We'll use our function recreated so it can work over multiple things, where we use pathview gene data equals full changes, spell it correctly, pathway ID equals PID, and then species equals mouse. Okay, and then let me close off this chunk and run it. Beautiful, so it is creating my pathview files for grader. So now, remember, we just need to take these two parts and paste them and change them to lesser, right? So we say lesser or less? Less. 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 Accidentally paste an extra R thing here. Alright. Okay, so these are less ones, right? So these are different cell cycle, thermogenesis, necroptosis. And so now, do I have to change anything here? Should be any full changes. Species mouse false. Keg res IDs. Let me think a second here. So this is written not the best way for cutting and paste, and let me show you why. So here, when we're making grader, we're saving keg res IDs, and we get this, right? This MU, and these were for the upregulated pathways. So then we plot them here, which is fine. Then we run this for less, and we save over our previous data. So we save over the grader with this less data, and then we run this. Here we should run it. Why is that? There we go. So now we've saved over keg res IDs, which up here was MMU03010, and now it's with the less pathways. So if I'm not going to go back and change this because it's a good learning lesson. But what I would do if I were to change this is for the greater section, call this KEGRES IDs greater. And then for this section on here, I'd call this KEGRES IDs lesser. And when you call them into the function to plot, use greater or lesser. Because you're losing your intermittent data. So if I have to go back, if I was like, man, I have to go back and look at why this isn't working, that data's gone, it's been overwritten. So just an FYI, it's probably bad practice, but it's a learning lesson. I can tell you why it's bad practice now and show you. Okay, so now we have plotted all these lesser pathways too. And so, excuse me. So what I want to show you though is how, when we use just an R script, they get plotted into a folder as a PNG file, and then you can open them up and look at them, right? That's great. But for the R Markdown component of this class, I want you to be able to publish everything in an HTML file on RPubs. And so you'll just show this code, but you wouldn't show your pretty files. So there is a way to actually load those files back up. So we're going to open a library called EdgeR. I wonder if I've installed this on this computer. Let's forget to put these last two here. Okay, so it is loaded. So it is installed. All right, so what I'm going to do now is take the file names from that directory, so the files, and your path is going to be your working directory. For you guys, it's going to be squiggly line desktop / classroom / my files. For me, I'm actually looking at this way too. So if your files are in the same directory you're working in, you can just go to get wd. So this is the directory I'm working in. So I'm just going to copy that and paste it. And then this will list all the files, but I only want the Pathview files. So I'm going to say pattern equals, and I'm going to use that regular expression that we learned. And I'm going to say dot star, which means anything before, and then pathview.png. So it's going to take everything that ends in pathview.png. So I'm going to run this line. And so now I should have file names, and it says I have 32. It's going to be a lot to list. So all images, I'll apply file names, load image. So let's run that. And then lastly, what I want to do, so it's loaded the image into the R environment, but it's not, you notice it's not displaying anything. So what we need to do is we need to tell knitr to include graphics from our file names. So when we knit this to an HTML, it will include all the graphics for these loaded images. And so now I'm going to say knit on save, and I'm going to save this and let it knit the HTML. It's a lot of graphics to load. I should have filtered out how many. I advise you to filter out and not include 30 images in your final, because it just seems like too much in your final portfolio chunk. But okay, so now if I look at my knitted package, I got my nice plot here, and then my volcano plot, which is great. I turn off these notifications too. Here we got the greater, so the Q value. So ribosomes is significant because it's different than 0.05 or it's smaller than 0.05. And then it's the rest of the stats. For the less, we see we have here's our Q value. Nothing. So nothing is significantly lower expressed in mice in space. And then we keep going down. I turn off all these notification warnings. But you can see you've now included all of your pathways and the gene expression. There's so many pathways. Okay, guys, I figured it out. The reason that I had so many files is because I forgot to delete them from the last time I ran this. So it's just going to load everything in that folder, right? So if you run this, you probably ended up with just 10, which is what you should. You can, see them. Oops. If I hit this, you should be able to see them. These should be the 10 that you have in your HTML document. So that is it. I spent a little bit of time in that edit figuring out why the heck I had 36 files or whatever. But yeah, so that command is just going to load anything with those names in it. So if you run this analysis over and over on different data sets, it's going to just take everything in, right? So that's why I had so many. So you should have 10 nice documents, nice images in your knitted HTML document. So that's the end of EdgeR. Happy to be done. We'll get into one more method, so I'm going to go over both the two kind of most popular methods, at least in my experience, which are EdgeR and DESeq. So we'll do the same thing with DESeq. We'll go through Pathview so we can look at what files are differentially expressed, and then in the video after that, we're going to, or after DESeq, we'll compare the results from EdgeR and DESeq and see where they're similar and where they're different. So I hope you enjoyed that, and I will catch you in the next one.